Hello, everyone. My name is Grady Mandel, and this is my biotechnology presentation on CRISPR. I work as a firefighter and EMT, and one of the calls that's always stuck out to me has been a sickle cell pain crisis. Watching somebody hurt that badly because of a problem written into a single letter of their DNA is kind of crazy to me. So, when I started digging into CRISPR, the technology that actually fix that letter, it was pretty interesting to me and grabbed me. So today I'm going to walk you through what CRISPR is, some of what it's being used for, and what I'd do with it if I ran my own lab, and the ethical side that comes with editing human DNA as well. So part one is, what is CRISPR? Um, what is it? CRISPR stands for clustered, regularly interspaced short palindromic repeats, and it's usually paired with a protein called Cas9, which is short for CRISPR associated protein 9. In plain terms, it's a set of tools that let scientists change an organism's DNA, adding, removing, or altering genetic material at a specific spot in the genome. It was first described back in 2012, and it caused a huge stir because it's faster, cheaper, and more accurate than the gene editing methods that have came before it. Here's the part that I think is wild. We didn't invent CRISPR from scratch. We pretty much borrowed it from bacteria. So bacteria gets attacked by viruses, and over time they developed a defense system. When a virus invades, the bacteria grab little snippets of that virus's DNA and file them away in sections called CRISPR arrays. That's basically a memory bank of past invaders. If the same virus or a close relative shows up again, the bacteria make RNA from those stored snippets to go find the matching viral DNA. And then they send in an enzyme like Cas9 to cut that DNA apart and shut the virus down. Scientists figured out how to turn that natural system into a tool that we can use. In the lab, researchers build a small piece of RNA with a guide sequence that's designed to match a specific target sequence of DNA. That guide RNA also latches onto the Cas9 enzyme. The guide finds its matching spot in the genome, and the Cas9 acts like a pair of molecular scissors and cuts the DNA right there. Cas9 is the enzyme they use most often, but it's not the only one. Other ones, called CPF1, can do the job too. Once the DNA is cut, scientists rely on the cell's own repair machinery to make the change that they're after, deleting a piece, adding a piece, or swapping into a corrected sequence, something like that. That's the whole idea essentially in a nutshell. Guide it to the right spot, make the cut, and then let the cell rebuild it the way you want. So that brings us into part two, which is the applications. When I'm talking about a sickle cell, but obviously there's much more that I can do. So what is it actually being used for? A lot of things. A lot of excitement is around single cell gene disorders, disease caused by a problem in just one gene, because those are the clearest targets to aim at. I think cystic fibrosis, hemophilia, and sickle cell disease all fall into that group. And beyond single gene diseases, researchers are exploring CRISPR for more complicated conditions too, like cancer, heart disease, and even HIV infection. The one I wanted to spend time on is sickle cell disease because it's a perfect example of how CRISPR could work. Sickle cell affects almost 100,000 Americans and a million more around the world. And here's the thing, it literally comes down to a single letter change in the DNA. There's one letter changed in the hemoglobin gene. Hemoglobin is the protein in your red blood cells that carries oxygen. That one tiny mutation distorts the hemoglobin protein. So instead of each copy floating around on its own, the copies stick together and form long fibers. Those fibers warp the red blood cells out of its normal round shape into a stiff crescent, sickle shaped, which is hence where it gets the name. And that shape is the whole problem. Red blood cells are supposed to be smooth and flexible so that they can slip through the tiniest blood vessels. Sickle cells can't. They get caught up and clog the vessels and block blood flow, which causes the severe pain that I've mentioned and I've actually gotten to see. And it means that oxygen isn't getting delivered through the body that it should be, and every one of those cells carries the same faulty instruction because red blood cells are made from stem cells in the bone and marrow. So how could CRISPR help? The idea is to go in and change that DNA to replace the mutated version with a healthy one so the body can build normal red blood cells instead of the sickled ones. Researchers, including the team at Innovative Genomics Institute, are working hard to turn that idea into a real therapy. And that's the promise of CRISPR in a single disease. Find the exact letter that's wrong and then correct it at the source instead of only treating the symptoms after the fact. So that takes us into part three, which is if I had my own lab. And if I had to do if I had my own research lab, what would I do with it? Given my background running firefighting calls and emergency calls, I'd want to work on the disorders that put people in crisis, and sickle cell would be right at the top of my list. Specifically, I'd focus on the delivery problem. It's one thing to correct the mutation in cells on a dish. The hard part is getting that fix into the right cells inside an actual patient and doing it safely at the same time. So I'd work on editing a patient's own blood, forming stem cells, the ones in the bone marrow that produce the red blood cells, correcting the sickle cell mutation in those cells, and then getting them to repopulate the body with healthy red blood cells. I'd also pour a big chunk of my resources into safety and accuracy, making sure that Cas9 only cuts where it's supposed to and not at some similar looking spots elsewhere in the genome, because a cut in the wrong place could create more problems than what we're trying to fix. So, honestly, in coming from an EMS world, I care a lot about access. If a therapy like this works, I'd want it to become something regular patients can actually get, not just a handful of the rich and powerful. To me, a cure would be, or a cure that can only be reached by a few people isn't really a cure at all. It needs to be mainstream. So that takes me to my last and final point, which is the ethical and scientific concerns. Uh, Now, the part you can't skip with this technology is the ethics. I think it's a slippery slope. Editing DNA to cure a disease sounds like an obvious good. And for something like sickle cell, I think that it is, but it gets complicated fast. And here's the key distinction: most gene editing is done on what are called somatic cells. That's every